Thanks, Nancy. And thanks everybody for sticking around. It's been an incredible day. It's been great. A lot of variation in topics, all great talks, and so random another switch in topics and talking about a whole different kingdom. So as Nancy said, I want to talk about fungal conservation, but really kind of why the importance of fungi, why it's important to conserve them, and what's going on in the process. But first, since I moved back, it's been almost 50 years since I was in Carbondale, and I just want to recognize Dr. Walter Sundberg, who was on the faculty at the time, who introduced me to the wonderful world of fungi, but actually to science. He's the first guy that really got me to do science and realized what the scientific process was. So I owe a lot to Walt. So this is a plant symposium. And, when I look at it, I do see the plants. I see the trees and the herbs and whatever else. But on the bark of the trees, I see the lichens. I can't see them, but I know inside the leaves and whatever else, endophytes that are providing drought resistance and pathogen resistance to the plants. For the grasses and things like maples, they're all obviously associated with these arbuscular mycorrhizal fungi who are bringing in nutrients and water, and again, providing some pathogen resistance. For the oaks and pines, whatever else, they're with a different group of symbiotic fungi with Ectomycorrhizae, and even the orchids get in the show, forming orchid mycorrhiza, which is essential for seed germination and for persistence. So the fungi play these incredible ecological roles, whether it's decomposers, recyclers, they're basically nature's recyclers, antagonists, pathogens, partners, and carbon sequestration. And I'll just mention that last one. This recent paper came out in Science Daily that we're looking at a number of papers that thinking that fungi are involved in about 30% of the sequestration going on. Might be a little bit of exaggeration, but everybody agrees that fungi are critical for carbon sequestration. So you got all these plants, the trees, whatever else, are sucking up the carbon. They're putting them down in the soil that's being taken by the fungi through their mycorrhizal partnerships, being either sequestered by them or actually put further into the soil and put into soil particles for long-term storage. So fungi are really, when we're talking about carbon sequestration, that forest and grasslands are doing, it's really the fungi and the other microbes in the soil that are putting that carbon into the ground and keeping it there. I'm just going to talk about one of those groups just because there's been a lot of public press right now about Ectomycorrhizas. And, this is the association between certain groups of fungi, really a lot of the big mushrooms and other fungi with things like pine trees or the whole Pinaceae, Fagaceae, so all the oaks and those relatives, eucalyptus if you're in Australia. So a lot of the big major forest trees. They're actually called Ectomycorrhizae because they modify the roots. Here we are. So this is one of these roots of this pine tree. And so this really is a combination of fungus and the tree root that actually causes various hormones that cause the roots to bifurcate or multiply. And these little tips here is one of my favorite fungus. It's Lacaria bicolor, which I did my master's degree on here at SIU. And this is just a cross section of the root, seeing where the fungi are on the outside of the root, and they permeate between the cortical cells. They'll permeate into the cells, but around that, and that's the site of nutrient exchange. So they take up the sugars, excess sugars for photosynthesis from the plant, and return with nitrogen, micronutrients, water, etc. And as I said, a lot of these are beautiful, incredibly gorgeous fungi. Some of them are tasty as well. And I always use this as an example of the importance of pure research, of kind of, so Ectomycorrhizae was this first reported in the late 1880s. And for 50, 60 years, the forestry world said, oh, isn't that sweet? Some trees form associations with fungi, isn't that nice? Until they tried to start planting trees in places where they hadn't been before. And so the textbook case is in Puerto Rico, where there were no native pines, no native oaks, or anything like that. So there are no native ectomycorrhizal fungi in Puerto Rico. So they went to California, got seeds of Monterey pines, brought them into Puerto Rico, planted them, and after 5 years, that's what the seedlings looked like. They didn't really do anything. So they went back to California, dug up the soil around that, the leaf litter or whatever else, and planted it. And this is what a 6-year-old pine looked like after they brought in the appropriate soil community, mainly the mycorrhizal fungi. So again, all of a sudden it went from, and this is 1960s, 1970s, so almost 100 years of it being a cute ecological oddity to being recognized as a essential part of forest ecosystems. And then further along, we've realized that, hey, when you grow these things, so this is basically soil, all of the cobwebby is the mycelium of the fungus. You can see the 3 kind of roots along here, and all the bright spots are mycorrhizal root tips. Okay? And the key thing here is you can see that this older seedling has a bunch of these, but when they plant the new seedlings, that this one mycelium, this one individual, can form mycorrhizae with these other seedlings. And so that led to some work where they were able, this is 1997, Susan Chimard's work, showing that there was actually net transfer of nutrients through these mycorrhizal connections from tree to tree. Okay? And so it was a big deal. And then a little bit later, there were some studies. This study was done on tomato plants. It was replicated on some other plants as well, where these tomatoes, when they're attacked by aphids, they give off a pheromone that attracts a parasitic wasp that attacks the aphids. But what they found was if you had these connected by a mycorrhizal network that and kept the aphids off of the adjacent tree, if this tree was impacted, it actually triggered a defense response from the other tree to attract the wasps. And so they were showing that there was some, again, communication between the affected tree and the non-impacted tree, which was going on. So, and that was actually documented. But since that time, this whole idea of kind of the Gaia hypothesis blown up, this whole idea of the, I guess they're now calling it the mother tree, has really taken off and probably overstated what's going on. So there's a lot of conversation now in the community on how much is going on, how much is the forest and grassland systems being driven by but what I'm trying to figure out, by cooperation versus competition, right? All of our ecological theory is based on competition, right? And all of a sudden, here's this whole idea that maybe cooperation is a major driver here. So there are kind of 2 schools of thought right now. Susan Simard is still sticking with her idea of this major cooperation going on, and others are really looking at those data. And we can talk about that more, but I would just say that there is unquestioned that fungi are critically important in the systems. Whether they're really driving everything is another question, but it doesn't lessen the role that fungi are playing. When it comes down to people, of course, a lot of people think about fungi from a food source. We just we're at the very, very tail end of the morel season. So a lot of people are hoping to find this, and everybody's hoping not to eat that. That's the deadly amanitas. So from a food source, it's there. This is some pictures I took in a Mexican market, and you can just see the diversity of fungi that are being sold and the amount. So from a food source, it's super important. 10 of the 20 most valuable human medicines are derived by fungi. The two statins of anti-cholesterol compounds. The first statins were from fungi. Penicillin had a little bit of impact. And the immunosuppressants that allow for organ transplant are all fungal based. And what's amazing is how recent in the Western world this is. So this is a thing on penicillin, the new wonder drug for mold. That was for, the Second World War. So this is 1940s. So we're only talking about 60, 70 years that we've really been thinking about fungi as a medicinal source. Where in Asia, this has been going on for thousands of years, and they've recognized that. Again, more modern ideas of fungi involved. So now psychoactive compounds, whether it's psilocybin, LSD, or other things, are starting to be recognized in their value in psychological treatment. And there's all these other new uses, right? So whether it is oil cleanup and environmental cleanup, reducing the challenge with colony collapse, or even creating packing material, building material, whatever else from fungi. And because of the important fungi are really becoming part of the whole kind of social milieu. This was an article in Vogue a couple of years ago where they were showing clothing that are fungal inspired. So this is Teriti's Versicolor inspired of that dress. And then, of course, there's this little TV show that came out that had something to do with fungi. And so fungi are really important. In fact, a group tried to get together an idea of how much of economic value fungi have. And so they estimated, as a quick back of the envelope, about $54.57 trillion annually is what the value of fungi are. So important critters. I think most people, and so it's not surprising that the FAO, the Food and Agricultural Organization of the United Nations, put out this post a couple of years ago that caring for the planet starts from the ground, and fungi are an important component of that. We also know that fungi are super diverse. I like this drawing that has critters drawn in scale to how many species there are. So we got these pretty inconsequential, from a diversity standpoint, of vertebrates with about an estimated 80,000 species, 74, so about 6,000 species that are, left to decide, be identified. Plants, about 400,000 species have been described, estimates 450. When we get to fungi, estimates of 2.5 million is the best estimate. They would go up to 10 million, depending on who you believe, but there's only 155,000 so far described. And then the invertebrates, insects and other invertebrates, with about 8.5 million. But if you take the percentage, fungi are the least known group of organisms, of at least eukaryotic organisms. Fungi are, even though I know I'm in a symposium, but fungi are more closely related to animals than to plants. They're the sister group to animals, but within the fungi, there's a lot of phylogenetic diversity and morphological diversity, ranging from things like single-celled aquatic chytrids to your red molds or whatever else that you find if you forget to throw the stuff out in the refrigerator. To these arbuscular mycorrhizal fungi I was talking about, to the higher fungi, the more further up on the phylogenetic tree, whether it's your molds or your morels or your deadly amanita. But it's also morphologically variable at any one group, right? So yeah, I work with mushrooms and related species, and we get all kind of variation from true mushrooms to stinkhorns to coral fungi to earth stars. And there's all kind of ecological variation from mycorrhizal things to saprobes to pathogens. So a lot of variation. and high alpha diversity. So high diversity if you go to one place. So this was an older picture, as you can tell, my hair color has changed over time. But this was from a 1/10 hectare plot, so about a typical quarter acre, so a typical suburban plot. And we in one morning, we pulled off over 100 species of fungi in this 1/10 hectare plot, the three of us. And if we go back again at a later time of year, we'd find a different suite of fungi. And if we went to the other side of the valley, we'd probably find a different suite of fungi. So it's not only that there's a lot of species, but in any one spot, there is a lot of species. And though this is all makes them important, it also makes them fun and challenging to work with. And so why are we finding all this diversity? Well, some of it is we're getting in field work. So this is Macrocebe titans. It's really not that heavy, but it is about 8 pounds, and it's over 1 meter in diameter. It grows out of abandoned leafcutter nests. So it's not the fungus that the leafcutters grow, but if you think of it as the leafcutters are doing is they have this big frass, they have their whole garbage dump there, and after they abandon the nest, there's all kind of resources for little critters, and this is one of them. But you don't have to go to Costa Rica to do this. This was a species of chanterelles that we described from Chicago, Cantharellus chicagoensis. And again, we kind of knew it was different, it was, but until the 1990s, before the advent of use of molecular tools, we allowed for a really broad morphological concept. We just allowed for a lot of plasticity and said that, yeah, it's just really variable. But once we got molecular tools, we could actually figure out what morphological characters were important for these different species. So we can just divide them up by DNA and then figure out how do they differ. And I'm just going to use our friend Morell's just because it's Morel season. And so before the advent of molecular data in Microbank, which is just a name bank, right, for fungi, the thought is that most of the species were in Europe, and all the discrete species were in Europe, and then North America and Asia had less. After they did the DNA work, realized, it's really Asia has the highest number of species, Europe the next, and the US, or North America next. And that there's high continental endemism. So really, we have only 10 species in common with 8 species in common with Europe and other parts of the world. And even in the US, there's high provincialism in North America, with the 22 species scattered about 7 in the east, 10 in the west, and only 5 in that intersection. And just for people interested, here's the 4 species of morels we have in Illinois. The dark ones, the angusticeps, are definitely gone. We might still have the tail end of America up there. I don't know, after it was 80 degrees less, now too. But I bring this up because when we had this idea that species, these large species concepts, and the idea that a species could occur on multiple continents across multiple different habitats, conservation really wasn't a big issue, right? Because if you wiped it out here, the same species occurred here and there and there. What we now know is that, like other organisms, most species of fungi have discrete distributions and discrete habitat needs and whatever else. So all of a sudden, conservation could be an issue. And in fact, we kind of knew that for a while. This is an article from Science in 1991 talking about the disappearing mushrooms, another mass extinction. The mycological community didn't have as good of a PR department as amphibians. But no, it's probably not as dire as frogs and salamanders. But still, we recognized way back when, 30 plus years ago, that fungi could be in trouble. And it shouldn't be a surprise because fungi face the same threats as animals and plants, whether it's habitat loss, which we've been hearing about, loss of symbiotic hosts, pollution, overexploitation, so non-sustainable harvesting, and climate change. So habitat loss, right, can be really extreme, like this is in Ecuador, where you can see the same pictures going on in Southeast Asia, where you have oil palm taken over, and here are the native plants. But, what happened in Illinois way back when, so this is between 1880s and now, but you don't have to go that drastic, right? So in Europe, there are these semi-natural grasslands, which are the place where these wonderful wax cap fungi occur. And they're now under threat because of change of management plans for these particular things, where they're starting to increase the agriculture by fertilizing them with nitrogen fertilizer and changing it, and they're losing these wax caps. Old growth forests, of course, are important, and there's a number of fungi that are only occur in old growth forests, but even in the Midwest, like our beautiful Shawnee National Forest, land management decisions have an impact on what fungi are occurring there. We also know that fungi are really sensitive to some forms of pollution, especially nitrogen deposition. So a study that I was involved with, and a study that some of my colleagues were involved with, showing that nitrogen deposition really reduces the number of mycorrhizal fungi in the system. And remember I said that, hey, mycorrhizal fungi are the ones that are really involved with carbon sequestration. So losing these species, changing those communities, may be having a really negative impact. And then unsustainable harvesting. Normal collecting probably isn't a problem, but there are certain species where this is a problem. So this is the caterpillar fungus, Ophiocordyceps sinensis, known from the high plateaus in China, Bhutan, and Nepal. And this is a little mushroom that parasitizes the larva of the ghost moth. And you can see the larva here, and here's the fungus coming up here. And it's one of these zombie fungi where it actually takes over the activity of the moth. It actually has neural compounds that does that. So the moth crawls up towards the top of the soil, goes vertical so that the fungus can be, the fungal spore carp can be produced up here and the spore is released. So this whole thing, is about this big, pretty small, but annually it's about 100 to 200 tons of this species is being sold on the market. And the reason is that the price in 2009 was up to $18,000 a kilo. So it's way more valuable than gold. And so some of these people in the area, that is almost their entire annual salary they make by harvesting these things. So there's this whole social aspect as we try to manage harvesting of these and still provide livable conditions for these folks. The other one I have up here is it's pretty interesting. In Thailand, they love this little earth star. Again, it's about this big, and you can see the amount here in this market. So some of these fungi are unsustainably harvested. Invasive plants. So I was up in Chicago for the last 40 years, and garlic mustard and buckthorn and things like that were our biggest problem. I've moved down here. I'm getting used to, again, to Japanese honeysuckle and other problems here. But in both cases, these invasive plants can have a negative impact on what's going on below them, right? So here's some studies that show that these plants like garlic mustard, whatever, that have allelopathic compounds are really impacting these arbuscular mycorrhizal fungi. Again, another one down there. In fact, so the plants and these invasive species can have legacy effects of releasing that, so that as it's changed the soil community and the compounds in the soil, it makes it more easier able for the invasive species to recolonize and more difficult for restoration. So all of a sudden, the invasive species live on, and part of the problem in our restoration is the loss of the appropriate mycorrhizal fungi. But the other thing can happen too. So there are some invasive fungi. So here's Amanita phylloides, the death angel, destroying angel, excuse me, and it's native only to Europe. But it was introduced into the eastern US in the late 1800s and into the West a little after that. And it's slowly extending along the East Coast and the West Coast and a couple other places. Some of you might have been following the case in Australia, Melbourne, a woman who killed her estranged in-laws. That was, but it was with this introduced fungus. And we also know that now from other things like Amanita muscaria, which is native around here, but it is being introduced in places escaping from pines, are replacing the native mycorrhizal fungi. Southern South America, Australia, places in Africa. And around here, we've got this thing, the golden oyster mushroom, which has escaped from cultivation. You can buy this at the farmer's market, but it's escaped from there. And as you can see, it can be pretty dominant. And the data are just coming out now to show that it's actually replacing some of the native decomposer fungi. And we still don't sure what all that negative impact is, but usually the native things work better than introduced. One characteristic you can see from these guys and these guys is that the things that we know are invasive are big, showy things. We don't know how many little brown mushrooms have been invasive or some of these other things. So there's probably a lot more that are coming in and causing potential issues. And then finally, climate change. We know that some fungi are changing and expanding their range. This is Amanita terzii that was originally described from Texas and is now up into Champagne, and I think a little bit further north in Champagne. So we can have direct effects like that, changing the phenology, changing distribution, and also indirect effects where you get asynchrony between the fungal response and their host response. And again, we see that warming, the good documentation that's changing phenology. We're seeing that climate change can be impacting reproductive success of fungi because we're seeing that it's changing spore size and things like that. So we don't know what impact that's having on reproduction. And importantly, even we're seeing these loss of fungal symbionts and compounded with the pollinator differences having a negative impact on, say, orchid species. So fungi are important. There's lots of them. They're having the same issues that other organisms do. So where are we in understanding what fungi are in trouble, what we need to do? Because the first thing we do is to kind of figure out what fungi are in trouble. We never have enough resources, never have enough time, never have enough whatever to just randomly choose fungi and say, I'm going to do something about that. But most fungi haven't been assessed, and that's really hindered our effective inclusion in conservation discussions, access to funding, decisions, and conservation action. And again, that slide I showed that was showing the figure on a relative to diversity. Here's a comparable figure for relative to the number of species that have been assessed and had their conservation status assessed. And you can see that for fungi, less than 4%, so less than 0.4% of fungi have been assessed. But even from that small number, we have some ideas. So this was a paper I was involved with 2 years ago, where we looked at some of the first species that were on the global IUCN list, the International Union for Conservation of Nature. We had about 600 species there, and we looked at what those 600 species, we found that about 48% of the species that had been assessed were in one of the threat categories. Were vulnerable, endangered, or critically endangered. Another 10% were near threat. So probably we should be paying attention to that. Now, we know this is an overestimate because of the biases in, we actually selected species to assess that we thought might be threatened. So we know that's a bias. And of course, we have data from, but to counterbalance that, we have data from North America and Europe and very little data from biodiverse rich places like Madagascar or Borneo and things like that. So there's probability counterbalance, but we know that there are species that are in need of action. If we look at North America and look at the NatureServe Explorer, use a little bit different system, but basically the same. They've got 13,330 species in their database. Only 1,800 of them have been looked at, and of those, 575 are threatened. We look at the IUCN list from North America only, there's 287 of 91. So if we look at both of those, that's still 30% of the fungi that have been examined are in some kind of need of conservation. And they're not little brown mushrooms. They're beautiful, big, beautiful, showy things, including some little brown mushrooms. In fact, Charlie was talking about ash tree. So the ash tree bolete here, it's one of these that are associated exclusively with ash trees. When the ash trees disappear because of the, the emerald ash borer, so does the fungus. So why are so few fungi assessed so far? What do we know so a little bit about it? You need a certain minimum data. You kind of have to know where they occur, what's the size of the population, how that population is trending. Is it stable? Is it getting bigger? Or is it declining? You need to know something about its biology. And then hopefully we don't just have a list of species that are in trouble, but we know what the threat is and some ideas of what the solution is, right? We don't want just a list of here's species that we need to do something. We don't know why they're breaking out. We don't know what we're going to do about it. But so the list hopefully includes those data. So what's our data sources, right? So we've got these incredible resources in natural history collections, millions of specimens in fungarium and herbarium. And it's now pretty easy to get those data. We have data aggregators. So this is the Mycological Collection Data Portal for the US, where you can go in and search all the fungal collections, almost all the fungal collections in the US, and get information from that. You have things like GBIF, the Global Biodiversity Information Facility, so you can get the data. But we still don't have enough information, right? Because natural history collections weren't formed to answer these questions, right? They were formed to document kind of a taxonomy and rough idea of distribution, but not fine scale distribution. Definitely not abundance. Nobody wants a million of a particular species, and definitely not tracking trends. So how do we get those data? Well, because there is so much discovery still there, it's really through engaging citizen scientists, right? So they're our best hope for documenting at scale what species are occurring, where, when, and how those patterns are changing. And so this is going on in other taxa as well, but this paper just came out, I think, 2 weeks ago, talking about the power of citizen science, giving some of the positives, but also some of the challenges and suggestions for moving forward. So it's a big topic. We're doing a lot of that work ourselves. A lot of people are on iNaturalist, and this just boggles my mind. This is something I just downloaded a couple days ago, or last week, I guess it was. For the US, there are almost 6 million records of fungi in iNaturalist. And what totally boggles my mind, 430,000 people are putting pictures of fungi up on iNaturalist. 430,000 people, where there's probably 15 people in the country that are employed to put names on fungi, right? So that gives you an idea of the value of citizen science. But we also know that the quality varies on iNaturalist photos, and there's not a even distribution of the fungi being done. So fortunately, in the US, we have two really good organizations that are helping with. We have the North American Mycological Association, which is basically a consortium of all the mushroom all the mushroom societies, mushroom associations, mushroom clubs in the country. They have about 12,000 members in NAMA. And then the Fungal Diversity Survey, which I'm pleased to be on the board of, is an NGO that's really focused on collecting high quality data and then getting that to practitioners. And so both these organizations are engaging people to go out and collect high quality data. More and more people setting up the DNA labs in their basement, in their garages, and doing DIY, DIY, do it yourself. Yeah, anyway, you know what I mean. Homegrown DNA sequencing. It's a long day, folks. DNA sequencing, and they're finding really cool things. And the last thing I just want to mention is using eDNA, so environmental DNA. This is where you go out and you grab a chunk of soil or roots or leaves, and you just sequence and figure out what's there. It has high potential, but we're still working out how to make it really best used in this. So we've got some great examples. Here's a study where they're using eDNA to kind of look at the conservation value of grassland areas in, I think it's just Scotland. This is some work on looking at Spanish cliffs and documenting diversity. So it works sometimes. Other times it asks really interesting questions that we need to think about. So this again is Brigida nobilissimus. Here it is here. If you don't know, this is Paul Stamets. Some of you may know that name. But anyway, it's this thing that can be like 3 meters across, one of the largest fungi around. The spore carp, the fruiting part of the fungus, is only known from about 113 punks. That's the total known population of that species. And it only grows on old growth spruce and fir. So trees that are, 200 years old and, this big around. So that's the business end of it. But when they did the eDNA, they started just coring all the trees around, and they're finding in almost all the different trees, little saplings and whatever else. So is it a rare fungus? The fact that it's there, the mycelium is growing in these things, but it can't reproduce except on certain trees after they're, hundreds of years old. We're not really growing a whole lot of 300-year-old trees right now. So, from a conservation, from a biological standpoint, it's really fascinating. From a conservation standpoint, what does that tell me that they're in all these other places that my guess is they'll never, ever develop and never reproduce and never add to that gene pool? So, those are the kind of questions that we're trying to deal with. Got a study of colleagues of mine in Sweden are doing this, and they're also finding that they don't find a lot of the rare species when they go out and sample. But you can also have false positives. So, this was in Chicago, and one of our problems is with these big-headed carp. And so we don't know that they put up an electric fence, that it's in the river, it's not in the lake. And so they don't want it to get in the lake because it's going to decimate the fishing population. So they've never found one of these carp in the lake, but they keep finding eDNA because they're getting sperm or, scales or whatever else. So they're finding DNA evidence that they're there, but they've never found the full critter. So again, you can get these false positives. You think of it from a fungal standpoint, spores blew out, you get the false positive from a spore, or the mycelium starts, but it's not the right conditions. Similar if you think of birds carrying seed from a plant, you can find seed all over the place. Doesn't mean that that plant is going to persist and add to the food. So anyway, we're still working on how eDNA is going to be used. But I guess my take-home point is between citizen science, the growing way we can mine data in collections and eDNA, we're getting to the point where we're going to be able to get lots of data. And we should be able to move on this. Oh, let's just say that we're going to do both. Never mind. But where are we? So for the environmental, listed in the endangered species list, there are only 3 fungi, 3 lichens. That's it. That's on the endangered species list right now. And I've done a survey of states for the state natural heritage programs, and I think one of them is starting to look at mushrooms. Maybe 2 or 3 are doing just capturing data on lichenized fungi, but not really doing anything with it yet. So we're getting the data, we're starting to work with them, but so far those data haven't been put to use at state level. And one of the things is we're trying to get people to recognize the value of thinking and explicitly thinking about fungi. So we've got the 3F project, the fauna, flora, and fungi. Fungi is the comparable world for flora and fauna. And so we're trying to get everybody to think about, when you say flora and fauna, you include fungi, or it's animals, fungi, and plants, and do that. And so we're starting to make some progress. Green Wild, which is a really big NGO, is doing it. IUCN is now including that. And just 3 weeks ago, National Geo had come out, and they've now included fungi as part of their definition of wildlife, and they had this whole volume of National Geo there. So we're starting to make progress, because unless you talk about fungi and include fungi in these things, it's not going to make progress. So just to conclude, we're making progress, but much, much more work needs to be done to document fungal diversity, their distribution patterns, conservation status, and then getting that work out there into conservation action. Because a lot of these data are observational, incorporating citizen science and improving the data that they're getting and putting those data in a place that people can get access to them is important. We're getting new computer tools to do that. And then while these threat assessments are important, the data need to be put to work. And that's the job of trying to get, whether it's Forest Service or heritage programs or whatever else, to include fungi into their work. And so my kind of statement I've been trying to use now with limited impact is that I think we're to the point with a lot of these agencies that they realize the importance of fungi, but then they say they just don't have the resources, they don't have they can't afford to include fungi. And what we're working on now is for them to realize that they can't afford not to include fungi. So thank you very much.